Let's talk about the nucleus. So as we know, the nucleus is going to be the housing for all of our genetic material. It's going to contain all of the cell's DNA, and then all of that DNA within our nucleus is going to play a role in the regulation or control of cell activities. And because all that DNA is in the nucleus, when we go to replicate our DNA, well, that's going to happen in the nucleus. So the nucleus is the site of DNA duplication, and all that DNA in there, um, you know, containing coding for genes is going to play a role in the control of cell activities. Now, within our nucleus, we have a region called the nucleolus, and so that's going to be within the nucleus, and that's going to be the region where your ribosomes are assembled. And we know that our ribosomes are composed of rRNA. Okay, so a little bit of the morphology of our nucleus. Our nucleus is going to be composed of what we would call a nuclear envelope. And as you can see here, it is actually doubly layered, kind of like the mitochondria. And it's perforated. So you got little holes in it. We call these nuclear pores. Now, just underneath the nuclear envelope, we have what we call nuclear lamina. And that's a meshwork of proteins that maintains the shape and integrity of the nucleus. So just inside of the nuclear envelope, you have that meshwork of proteins called the nuclear lamina. And then the envelope is the envelope itself. So the membrane around the nucleus, which is perforated with nuclear pores. So your nuclear pores, those are going to be the structures that help connect the interior of the nucleus to the outside of the nucleus, or the cytoplasm. So it connects the interior of the nucleus with the rest of the cytoplasm, and those holes, or those pores, allow things to move into the nucleus or move out of the nucleus. So RNA and proteins must pass through these pores in order to leave the nucleus. And then embedded in these pores, you have a pore complex. So it's just a complex of different proteins called nucleoporins that surround each pore. So a lot of these have to do with filtration. They're going to filter things out of the nucleus, or filter things from leaving the nucleus if they're not supposed to. So here's just kind of a general overview. Some of the anatomy here. This is your nucleus. Here's the nuclear envelope. That's what NE stands for, nuclear envelope. CF just stands for cytoplasmic filaments, and then NF stands for nuclear filaments. And then your nuclear pore complex, that's what's encased right here. So here it says selectivity filter. So you're going to have complexes of proteins that help with selecting what goes in and out of the nucleus. So if you look here, you've got the little green dots. Those are small neutral molecules which can pass through pretty easily into and out of the nucleus, whereas large neutral molecules, well, they have to have some kind of signal in order to enter or exit the nucleus. So small molecules can probably move through, but whereas large molecules need some kind of signal. Okay, so now let's talk about transporting stuff into the nucleus. How do we get stuff into the nucleus? Before we jump into the mechanism, let's talk about a little bit of terminology. The first term is nuclear localization signal. So that's going to be a peptide or protein tag, usually consisting of positively charged lysines and arginines. So like a sequence of lysines and arginines embedded into the protein cargo that you want to bring into the nucleus. It's going to have a tag embedded into it. And the next we have a term called importin. So importin is going to aid in the import of cargo into the nucleus. So it's the transport molecule that brings other molecules into the nucleus. Very important. And then lastly, we have a complex called RAN-GTP. RAN just stands for RAS-related nuclear protein, which you don't really have to remember, but it's going to be bound to GTP. So kind of like ATP, GTP is also another type of potential energy we may use within the cell. So RAN-GTP is a complex that helps importin. Remember that importing molecule, that molecule that is very important in important molecules, it helps important dissociate from the cargo so that we can let that cargo free and that cargo can do what it needs to do within the nucleus. So it helps important dissociate from the transported molecules. So now let's go over the mechanism. So we have a protein that contains a nuclear localization signal. It's embedded in the protein. And that's going to have an affinity for important binding. So important is going to see this nuclear localization signal. It's going to bind to it. It's going to have an affinity for binding to it. And then what's going to happen is that protein bound to important, so that complex will have a higher chance of actually being brought into the nucleus. So it can pass through the nuclear pore filters, the filtration process. It now has signal or a carrier to bring it into the nucleus. So that important protein complex can now move into the nucleus. Once it moves into the nucleus, the protein important complex enters the nucleus, and what's going to happen is it's going to bind to what we would call RAN-GTP. So RAN-GTP is just another complex, but what this complex is going to do is it's going to take away important. So it binds, so this RAN-GTP actually binds to important, relieving the cargo to do whatever it needs to do. It's no longer bound to important. It doesn't have a carrier bound to it anymore. And now it's just going to do whatever it needs to do within the nucleus. Now we're left with this complex of RAN-GTP. So the RAN part is the purple thing. The GTP, well, that's just the GTP part. And then that green thing, that's the darker green, that's going to be the important. So now we have this trio, this RAN-GTP bound to important. That's going to have an affinity to be transported out of the nucleus. So this important RAN-GTP complex then leaves the nucleus and then eventually becomes hydrolyzed through an enzymatic process. This GTP is hydrolyzed down to GTP is hydrolyzed down to GDP, which causes the dissociation from RAN-GDP and important. Now important is now recyclable, and it can be used to bring other cargo into the nucleus, whereas your RAN-GDP can actually be brought back into the nucleus and then have a phosphate group added to it so that it can be recycled as well. So important plus RAN-GTP complex moves into the cytoplasm, and then the GTP is hydrolyzed to GDP, important dissociates. So that's how we get stuff into the nucleus. Now let's talk about how we get stuff out of the nucleus. So some terminology before we go over the mechanism here. Exportin is going to be very important for exporting cargo. So it's going to be a transport molecule that carries cargo outside of the nucleus and into the cytoplasm. And much like your nuclear localization signal, what we have is a nuclear export signal. That's going to be embedded on whatever molecule we're going to want to bring out of the nucleus. So your nuclear export signal is going to be, again, it's going to be a peptide tag or a sequence of amino acids, usually consisting of hydrophobic amino acids. So it's going to have a chain of hydrophobic amino acids, which has an affinity for particular molecules like exportin. Anyways, our last term here is RAN-GTPase activating protein. All you have to remember is RAN-gap. RAN-gap. So this is going to cause RAN-GTP to hydrolyze into GDP. And that's what's going to cause the dissociation of everything once you get it out of the nucleus, you could say. So it causes RAN to release exportin and the cargo protein into the cytoplasm. All right, so let's go to the mechanism. So exportin, that's going to be an important protein, or you could say molecule, in exporting goods from the nucleus. That has an affinity for the nuclear export signal. Now RAN-GTP, that should look familiar. RAN-GTP is also going to be involved in transporting things out of the nucleus. So let's say you have that nuclear or that protein containing the nuclear export signal. What's going to happen is exportin is going to bind specifically to that nuclear export signal, and then RAN-GTP is going to bind to exportin. Now you have this trio, or you could say quad. You have the GTP, you have the RAN, you have the exportin, and then you have the cargo protein with the nuclear export signal. All of that can now be transported out of the nucleus. They can bypass the filtration process of you could say the filters of those nuclear pores, and they're brought out of the nucleus into the cytoplasm. Now, just as they leave the nucleus into the cytoplasm, there's going to be another protein that will aid in the hydrolysis of GTP on RAN-GTP. So RAN-GAP will cause RAN-GTP to become RAN-GDP. So that's a guanosine diphosphate. And what that does is it causes the dissociation of the cargo protein. So RAN-GAP is going to hydrolyze or help hydrolyze RAN-GTP into GDP, which then causes the dissociation of the cargo protein from everything. So then what you're left with is exportin, you're left with RAN-GDP, and you're left with RAN-GAP, which just stays in the cytoplasm, and then that cargo protein, which is free to do whatever it needs to do outside of the nucleus. Now that exportin can be brought back into the nucleus for recycling. So that's how you get stuff out of the nucleus. All right, so speaking about the nucleus, we still have a few more terms that we want to know about the nucleus before we move on to another cell part. We have chromosomes. So chromosomes, that's going to be the form of our genetic material. So each of our linear pieces of DNA, we just call those chromosomes. All of our chromosomes, we have X amount of chromosomes if you're a species that has linear chromosomes. Your chromatin is actually going to be a condition of the chromosome. So your chromatin is actually the chromosome in its decondensed, uncoiled form. So decondensed and uncoiled DNA-bound proteins. So chromatin is going to be a complex of nucleic acid as well as proteins. If you remember, the DNA is going to wrap around these little proteins called histones. So it's just going to be a complex, and it's not necessarily tightly condensed like a condensed chromosome, which we normally think about when we think about chromosomes. They're usually the bilobed condensed chromosomes. When you're in interphase, all of your DNA is in chromatin form. It's all decondensed spaghetti like this. And then lastly, we have nucleoplasm, and that's just going to be the liquid suspending all of the chromatin inside of the nuclear envelope.